Welcome to Lecture 13 on the topic of plant signals. To complete this topic you will need to watch all 3 lectures. This is the first part. This lecture is a module of the plant physiology subject, which is a component of the Bachelor of Agriculture and Technology offered at North Melbourne Institute of TAFE. Please visit our website at www.nmit.edu.au for further information on both this subject and other courses that we offer. My name is Dr. Nikki Cooley. This lecture is being taught in Stage 2 of this subject. Stage 1 allowed plant physiological concepts to be taught as a virtual tour through the plant. This stage allows us to look at plant signals both across a whole plant perspective and at a greater detail than we were examining topics in Stage 1. Plant signals involves hormones and compounds that can be used in communication, both within the plant and across to other plants. Plant signals are also used in defense and in some industries are important as they are a quality component of that industry. So what are we hoping to achieve in Part 1 of Lecture 13 on plant signals? Well, I'm going to introduce hormones and hormone classification. What are hormones? What are the most popular groups? Then we are going to talk about one of the most important plant hormone groups, that is of auxins, abbreviated to IAA. You'll be introduced to the structure of auxins. You will look at some of the functions of auxins and the biosynthesis, and how this hormone is moved around the plant. We will start this topic on plant hormones with a definition of a hormone. A hormone is a chemical messenger produced in one cell that regulates the process in another cell by interacting with protein receptors which are linked to cell pathways. Chemical messages are required for the efficient communication among cells, tissues, and organs in plants. In plants, regulation and coordination of metabolism, growth, and morphogenesis depends on chemical signals. There are 6 major categories of hormones in plants. These are auxins, gibberellins, cytokinins, ethylene, abscissic acid, and brasinosteroids. There are also other signaling molecules such as jasmanic acid, salicylic acid, some small proteins, stritoglatone, and flavonoids. So let us explore each of these classifications of hormones in more detail. It is perhaps fitting that we should start with auxin, as this was the first plant hormone discovered. Auxin has been found to function in every aspect of plant growth and development. Auxin works alongside a cytokinin, and these hormones differ from other hormones as they are required for a variety of plant embryos. In the 1800s, Charles and Francis Darwin studied plant growth processes. This was called tropism. The Greek tropos means to turn. This process, caused by differential growth, is called phototropism. Using the canary grass seedling, the interaction of auxin and plant growth and development can be explored. After emergence from the seed, the canary grass has very young leaves, and these are sheathed or protected by an organ called the coleoptile. The coleoptile is highly sensitive to blue light. If you illuminate on one side, the coleoptiles will bend or grow towards the light source within an hour. The tip of the coleoptile perceived the light, for it covered with foil. The coleoptile would not bend. Therefore, it was concluded from this research that some signal is produced in the tip, travels to the elongation zone, causing the shaded side to grow faster than the illuminated side. This observation was published in 1881 in The Power of Movement in Plants. It was experiments conducted by Fritz Wentz in 1926 on the tips of oak coleoptiles that went some way towards the discovery of Auxin. He enabled a major breakthrough in the isolation of the chemical with his clever experimental design. He allowed the chemical to diffuse out of the excised coleoptips directly into gelatin blocks he had placed. The gelatin blocks were placed on one side of the decapitated coleoptiles, and they could be tested for their ability to promote bending in the absence of light. This substance we now know as auxin. The visual representation on the slide shows a summary of Wentz's experiments. It also shows some of the results and the conclusions that he was able to draw. Please watch the following video on Wentz experiments called Phototropism and Auxin. This will help you visualize the results from these experimentations. Auxin was discovered in the 1930s. The major natural auxin is called indole 3-acetic acid, or IAA for short. There are other auxin forms that exist, for example 4-chlorine IAA, which was discovered in peas. IBA was discovered in maize and legumes. The synthetic auxins used in herbicides in agriculture and horticulture are called 2,4-D and Discampia. 2,4-D was released in 1946 and was the first selective successful herbicide. It kills broadleaf plants, not cereals including wheat, rice or corn. It causes uncontrolled, unstable growth. Discampia causes uncontrolled growth in plants until nutrient supplies are exhausted, and then the plant dies. The biosynthesis of auxin is often associated with rapidly dividing and growing tissues, especially in shoots. The shoot apical meristem, or SAM, and young leaves are the primary sites of auxin synthesis. Root apical meristem, or RAM, also synthesizes auxin, although most of the auxin derives from the shoot. IAA is related to the amino acid tryptophan, which can act as a precursor for IAA biosynthesis. Auxin, or IAA, moves from the main apical to the basal end. This is called basopedally. This unidirectional transport is termed polar transport. The image on the slide summarizes the movement of IAA. Auxin was discovered as the hormone involved in the bending of coleoptiles towards light. This results from unequal rates of cell elongation on the shaded side versus the illuminated side. Auxin is transported basopedally in the vascular parenchyma of the shoots, then moves laterally to the outer shoot tissues. The process of lateral auxin transport is mediated by PIN transport proteins. Auxin can cause a staggering 10-fold increase in plant growth rate in only 10 minutes. So how does it do this? Well, firstly, we need to review the role of cell elongation. Osmotic uptake in water driven by water potential gradient. Turgor pressure increased due to rigid cell wall. Cell wall loosening occurs, then allowing expansion. Auxin has been found to increase the wall extensively by increasing the rate of proton exclusion or wall acidification. This increases in acidity promotes the activity of expansion proteins, which then loosen the cell wall. Auxin is involved in the actions of 3 plant tropisms. Phototropism, which is the growth with respect to light is expressed in all of the shoots and roots. It ensures that leaves receive optimal light. Geotropism is growth in response to gravity. This enables roots to grow downwards and shoots to grow up. Critical response during germination. Thymagtropism is where growth with respect to touch enables root to grow around obstacles and the ability of some shoots to wrap around structures for support. When dark-grown oat seeds are orientated horizontally, the coleoptiles bend upwards in response to gravity. Auxin is transported laterally to the lower side, causing the lower side to grow faster than the upper side, and this explains the mode of action of gravitropism. So how do plants detect gravity? Well, the main candidates are the large, dense aminoplastids or starch-containing plastids. They are highly dense and relative to the cytosol, so their sediment falls to the bottom of the cell. The aminoplastids that function as gravity sensors are known as statholiths. Auxin has many developmental effects. These include apical dominance, floral bud development, leaf arrangement, lateral root formation, vascular development, leaf abscission, and fruit formation. In most plants, the growing apical bud inhibits the growth of the lateral buds. This is known as apical dominance. Removal of the shoot apex results in the growth of auxiliary buds. Auxin can stimulate the apical bud in maintaining the inhibition of lateral buds. Classic experiments were performed on kidney bean plants in the 1920s to demonstrate apical dominance. The figure on your screen illustrates the apical dominance experiments, showing the terminal bud intact, the terminal bud removed, and auxin added to stems that had been decapitated. Please watch the following YouTube video on the topic of auxin. This resource talks extensively about apical dominance and the effect and the role that auxin plays in this response. It is very important that you read the associated literature with this topic. Please read the Tays and Zeiger Plant Physiology 5th edition, Chapter 16, the section on hormone signalling, page 364 to 366, and the entire Chapter 19, Auxin, the growth hormone, pages 426 to 459. Please make relevant notes and insert your reading at this stage of the lecture. So to summarise the first part on the lecture on plant signals, you should now be able to define a plant signal and understand and define what a plant hormone is. We started this topic on the plant hormone auxin, a very important plant hormone. So why is it an important plant hormone? This is something you should now be able to address. What are the functional roles that this auxin plays in plants? And how does it go about ensuring that these functions happen? You should be able to describe some examples of auxin. Please ensure that you do the associated reading. This brings us to the end of part 1 on lecture 13 on the topic of plant signals.